So please ask your questions. Let me refresh here. I'm going to start with one question, which is really to Rong, which is, we talked about simplicity, and when you look at all that clinical data, it's extremely complicated. So will computers ever read charts for us and be able to structure that data without humans intervening? Is NLP or some other computer program going to read a chart and put the data into a format that we can use it?
Yeah, the short answer is we need both. We need computational tools to turn it into a structured forms and in use in daily clinical activities. And so first one is whatever we using NLP, we kind of structure there. And second, we need to show the original physician notes with original sentence, so you know why we make a decision to help you to decide whether you take it or change it. And we need the manual curator to kind of look at this and deciding to change or not. So that's one. Second is only through this iterative process, you're getting better and higher accuracies. And I would say is we have been shown for lung cancer, we've been able to fully automatically kind of writing the script to do what I showed, but we are looking that need extra manual curation as well as multiple different sources to figure out the accuracies and how we improve the accuracies. And second is in terms of accuracy, there are two kinds. One is at the cohort level, one is at the patient level. I think at the cohort level, that's much easier, but at the patient levels, we have to see because it's really a lot of one, still through manual curation, you know the exact truth. But overall, I would say it's just we have to do it iterative to continue improving the accuracy.
Thank you. This is a question for Pei. Are histones included in the proteomic analysis of tumors? And what about post-translational modifications of histones or other proteins?
Not right now. We are only focusing on the phospho, and that probably is the most ready kind of PTM right now.
Okay, and there was another question about how you decided on the tumors you chose. Some people asked about hematologic cancers, but there's a whole bunch of other cancers you excluded. How did you choose those 6 cancers in this current phase?
Okay, that is a good question. I think the main driven factor is how fast we can collect enough sample for those tumor types from our network. So we require treatment-naive tumors and with certain size. And for some tumor, that is particularly difficult than other tumors. So that's how we picked the 6.
I also thought it was unusual that there are 5 different data analysis centers. Is that because the NCI has no confidence that any of you are going to have the right way to do it? I've never seen that before. There's only 3 centers that collect the tumors and 5 centers that analyze.
That is because in the last cycle, we realized it took longer for the data analysis than the data generation. Therefore, the NCI thought, okay, then we should put more manpower to that stage. So that's why they selected really 5, funded more data analysis center than profiling center. And so we are, the 5 data analysis center are working very closely. I would say that we have different strengths with different type of data, with different tools. So I think that really benefits.
One question for Boris. What cancers have the strongest prognostic group differences in your experience?
The answer, which of the cancers have the biggest difference in, one more
That's
time?
Ramon's question.
These molecular signatures for prognosis, based on how you showed, which ones rank the best at being able to separate very good prognosis group from poor? Because some cancers, if they're very homogeneous, you won't be able to do this. Other cancers, it may split into 4 or 5 groups, etc. I just was wondering what your thoughts were on this.
The expert answer is probably lung cancer.
You have to write it in there, Miriam. I'm not allowed to call on you. You have to type it borrow Ramon's phone. So another question, this is let me ask Zainab. You know, when you see the 3D visualization, for those of us who are not looking at these really large data sets, it makes a really nice graphic, but what exactly do you get from that kind of 3D visualization? What, as a quantitative scientist, what are you looking for when you put all those genes into those beautiful figures and spin them around?
Well, we have another dimension. So when we have a really complex network or a very large network, then when we look at it, it's a hairball. However, we can, through innovative layout algorithms, we can make it understandable and communicable with other researchers, and that's the most strength that we have. And for example, the individual who generates the data can also be inside the network that they're looking at, and you can directly interact with a specific nodule. And all you have to do is either rotate the network or just move your head a little bit to see where they're interacting with. Yeah. Okay. With 3D. Yeah.
All right. So yeah, I think I saw an exponential difference in one of those slides of e to the minus 300, right? It was like 10 to the minus 300. So I think when you see those kinds of big differences, I'm not sure what you're visualizing and how it can help to understand this. One question is, what is the rate of success of big data analysis versus other modalities in identifying tools? This is for you, Ben. And either biomarkers or targets that ultimately make it to the clinic. In other words, you do all of these big data analyses, you have very complicated dilution. What actually makes it to actual practice, in your opinion, or what have you shown?
Yeah, I think this it can happen through these initiatives. I think the idea is really bring together this clinical data Ron is collecting, and also the data actually Pei is generating, and then we can go through this kind of multi-scale network models to identify patient subtype, and also identify mechanisms for each subtype, and then we can predict drugs for each subtype, and eventually, if we have single-cell data, hopefully we can predict drugs that can target those subpopulations of cells resistant to disease. And this kind of procedure, I think this is exactly probably Dennis and Icahn organizing now.
Okay, Boris, you wrote several questions yourself, even though you're on the panel. And I know you did it yesterday, but I'm going to give you a chance to maybe address something, which is, it sounded like you were asking, and all of you are quantitative scientists, you want your targets to be translated. You want to basically follow up on it. And I sensed in your question some frustration about how you can actually take this quantitative data and turn it into something real. Do you want to maybe address that concern? It was a question, but it sounded more like a comment.
Yeah.
Speak into a microphone, thanks.
Okay. I will just maybe repeat a little bit what I was talking about. That we have, when we started this approach, we didn't know where will we come. But currently, we have a framework which makes possible to analyze, first of all, TCGA data. And we find lots of very interesting findings. We can classify these cancers, we can find the difference, we can map the difference on the network. We see that we find very interesting genes with some biological, and we can easily make a biological hypothesis of what's going on. So then, of course, we can publish this, and it became very interesting. But now I will just make a link to what Dennis told in the morning, that we want our work to be really to make a difference in cancer. So first of all, we need more data to validate, and maybe specifically if, for example, like in this kidney cancer. And then if it's validated, we want to, that it will be used in clinical practice. And here we have a big question. I don't know how to move it to the clinical practice. Actually, for ovarian cancer, we have some signs, some very small data sets, of course, that our predictions are validated. All this analysis was done on one data set, and then these disease subgroups were found in another data set.
I think to summarize that point, and really this purpose of retreats like this, and maybe for the leadership of both the Icahn Institute and the TCI, is what structural ways do we bring quantitative data scientists to biologists who are sitting in the audience, clinicians who are sitting in the audience, to facilitate this? Because as I agree with you, in the end, everything's often one-off. People try to find out who's doing this disease, and we try to work on a one-on-one basis, but I think that would be one of the take-home challenges of this retreat, Ramon and Adam, to figure out a structure for people to kind of, like a clearinghouse for matchmaking people with targets and clinical interest or biology. I still don't see your question, but I'm going to get to it because I have the last minute for that. So there's a question here from Andrew. From Andrew Kasarskis, I think, for Ben and others. What are your thoughts about the optimal balance of single cell versus bulk tissue when you consider the cost and value of the data produced? So single cell sequencing is a very hot area, of course, but a lot of the genetics and genomics look at pooled data as well. So which is the right approach? Whoever would like to take.
Yeah, actually, we are integrating both bulk tissue sequencing data and also single cell. For single cell, it's easy to identify those subpopulations. I think that give you a more precise mechanism about like chemo resistance, and that's what we observe in breast cancer and also ovarian cancer. That's something actually you cannot really see from the bulk tissue, but on the other hand, for single cell data, won't be able to tell you much about the interactions between actually cell types. And the bulk tissue has the advantages. So I think that the best strategy would be to put together two types of data and do integration. Yeah.
Okay, thank you. It seems like they're both going to be around for a while. Yes. Zainab, somebody asked, are there efforts to create a best practice guideline in data analysis? Seems everyone has their own methods.
This is for the exome sequencing project, I
think
assume? In our study, we use the GATK best practices because that's the most friendly, reviewer-friendly when you're publishing. But when we do that, we also have to have a complete pipeline of analysis because there's the quality control step and variant filtering steps. And for all those, we, of course, have to build our own pipeline. And in addition to that, when we use GATK, there are a lot of steps, and thanks to individuals at Icahn, we already have a pipeline that does that with GATK, so it makes it easy, relatively, but most importantly, we can run thousands of exomes in our high-performance computing cluster, and to be able to do that is great on its own. So we do follow the best practices whenever possible. Yeah.
Great, thank you. And I did get the last question for Nina and Miriam before it beeped. And this will be for Pei. How do you account for tumor heterogeneity in proteomics analysis?
That is a very good question. So in CPTAC, we are doing bulk tumor profiling. And for many tumors, actually, this tumor heterogeneity has been quite an issue. And specifically, right now, we are more address the cell type heterogeneity in the current protein and RNA profiles. So this is a very active kind of data analysis and research topic that we have been working on with our current tumor data. Like, for example, in this kidney tumor, we are particularly care about the immune cells infiltration, and there are multiple computational tools in order that can help us to identify, say, the percentage of different immune cell types in each tumor, and we have already observed very interesting signals that those information seems to captures the, say, the stage information of the tumor and so on. So yeah, that is a very active research topic, actually.
Great, thank you. Thank you very much to the speakers for an excellent session. The next session starts at 1:10. Please be back here around 1:10. For the speakers on the next panel, please be here at 1 o'clock to set up. Lunch is on the 3rd floor in the library. Okay, thank you.